All right, so we're going to talk really quickly about microscopes, or the science of microscopy, and this is how we study cells. Okay, so notice cells are different sizes. These we can see with our naked eye, what we call the unaided eye. These we need a light microscope, which is what we use in our lab. So it's a more simple, doesn't magnify as high. And then we have an electron microscope to see smaller things. All right, so this is just showing you some different pictures, sizes. So notice the scale is much more zoomed in compared to these. All right, so when you compare them with a light microscope, light is passing through the specimen. It will refract light, and that will magnify your specimen, and it can magnify up to 1,000 times. So our light microscopes, the eyepiece is a 10x, and the highest objective is 100x. And so you multiply those together to get your final magnification. And so if we were using the Ohl immersion lens, which is that 100x lens, we could magnify up to 1,000x in our labs. The specimen can be alive and moving or dead. We can look at it in real life because it can be alive, so we can see it in color, and the light will allow us to see that, or it can be stained. And the electron microscopes are a little different. They require you to focus a beam of electrons through the specimen or onto the surface of the specimen. They magnify a lot more, up to 1,000,000 times. However, because the electrons are going to be hitting the specimen or going through it, if the specimen wasn't already dead, it would be killed. So the specimen is always non-living and in a vacuum. And sometimes they're frozen or dipped. I know that I think it's the scanning electron microscope, they have to be dipped in pure gold, so that can get pricey as well. And they do give us pictures in black and white. The only way to add color to these is to add the color after the fact. So you're basically not photoshopping, well, maybe that's what I want, but you're touching up that picture after the fact and adding that color to allow the person to see the differences. But everything is in black and white with those. When you're looking at the two types of electron microscopes, to transmit means to go through. So these electrons are going to go through. It's going to give us a 2D image with a lot of detail, and it's going to allow us to see the inside of the cells. With a scanning electron, it's going to scan the surface, which it kind of gives you a really cool picture. It gives you a 3D picture, but you're not going to be able to see inside the cell. So it really just depends on what you need to see in the cell as to which one you would use. These are scanning. This is actually an ant. This is the antenna of a moth. These are two different kinds of pollen. And this is a fungus that's budding off. And this is transmission. And you can see the cell, you can see the nucleus, the nucleolus, but you're seeing inside the cell. The newest microscope, and I use that term loosely because it was the 1980s when it was invented, is the scanning tunnel microscope, and it gives us pictures all the way down to the atom. So you'll notice it wasn't on that earlier picture that told you what each one kind of size-wise, because the book probably just hasn't added it yet. And so this is showing you. Now, I can't tell a whole lot about this picture, and so the people who do know a lot about the picture have added these, superimpose these images. And so you can see these are actually carbon atoms, those rings, and then the bond that's holding them together. And so it looks kind of like a honeycomb, I guess you could say, but what we're actually looking at is carbon rings. A couple other methods, because we do use other ways to study cells beside microscopes. We can fracture the cell, so we can break it into its fractions, to its parts, and then we can centrifuge it to separate it. And so, with centrifugation, we spin it really quickly, and the heavier stuff goes to the bottom, and the lighter stuff goes to the top, and so we can separate our organelles that way. So those are just some other things that we can do besides microscopy, but microscopy has given us the most information on cells throughout the years. And so that's a little bit about microscopes. I hope that helps. And if you have any questions, let me know.